Hi, you have this is Sadashiva Pai from Science Mission. We have Dr. Avidor-Reiss with us today. He is the professor in the Department of Biological Sciences in the University of Toledo at Ohio, United States. So, Dr. Avidor-Reiss has won several awards and grants. I'm going to mention only a few of them because it may take forever to do all the grants and awards he has won. In 2016, he won NHRO3. So in 2017, he won faculty member of the Council of UNESCO International Cell Research Organization. In 2018, John D. and Julius Jacobson endowed professorship in biomedical research. He's also editorial member and reviewer for several journals. Welcome, Dr. Can you please talk about your background, education, and some of the work you are doing, please?
Yeah, so my background is that I really did a lot of transition through my career. So I really start with asking questions in my very early education about vesicular transport and fusion. And then as I move up with my career, I move more to ask questions of opiate addictions. And then I really did a big transition to fly genetics. And as you're going to hear today, we're actually now moving forward to ask questions that are related to the human condition. And hopefully we hope to move into more clinical aspects. So we are really doing from very basic mechanisms to kind of a general, more clinical aspects.
And can you please talk to us about your lab people, grad students, and all?
So the lab is based on PhD students. Right now we have 5 PhD students. They work closely with many undergrads, so our papers are usually a time of a teamwork. We also collaborate with a lot of people outside of our university. Our research is very collaborative, and we are really benefiting from working with many other people in the scientific community.
Okay. And how do you motivate them? Because these are all small kids trying to get their research going. As you know, research is up and down all the time.
Yeah, well, one of the things that people that were in science for a long time can do, they can put in perspective. And I think that's what needed to be done. We do a lot of things. It's, the key is to keep it exciting. The risk may go up and down, but you collaborate with other people, you interact, you write reviews. If things are down, write a review about the research. So we do many things. So when things don't go very well to your own project, you are also working as a team. So people have their own project they lead, but they also participate in a team of other people. So based on where are you in your project and how things are going, we are kind of making sure that you are all the time engaged and there are something positive going on.
Okay, that's good. And can you please talk about some of the role models and mentors in your career?
So in my career, I had actually 3 very different mentors. In my master, when I did my master, I had a mentor, Dr. Daphna Atlas. She was really, it was fun because with her, we just speak for hours on models and ideas. And it was very exciting time as a young person. Then I moved to do PhD with Dr. Tzvi Fogel in the Weizmann Institute in Israel. And he was really a guy that he was very strategic. He did things very organized. We were able to publish many papers at that time because he was such an organized guy. And he always was thinking about how do I make sure Tomer career is okay. So I learned from him how to look on the projection of where the students will go and make sure that they are moving ahead correctly. And then I did my postdoc with Dr. Charles Zucker at UCSD. And this was really a very exciting time because this lab really made many major discoveries on different things. And I learned from him also to look on really on the high impact things. You want to make a high impact finding, take a high risk because there is a high gain. Don't only work on what is a sure bet.
Yep. And coming back to your, I know you are busy with all your research and all those things. How do you have this work-life balance? What are your hobbies and other interests other than the research?
Well, I don't have too many other interests, I must admit. I
Okay.
really love science. I
Uh-huh.
do have a hobby that is kind of heard some little bit strange, but I really like to read about history,
Uh-huh.
specifically on the Iron Stage in the Middle East. I'm really excited by this transition in the human history, and I read a lot about this archaeology. But I really more kind of, I would say, quite a single-minded person. Science is my top priority.
Okay, that's good. So now let's talk about your research and how do you get into all this developmental biology? And Charles Zucker Lab does a lot of developmental biology stuff. So can you please talk about your research and present your work?
Yeah, so a key thing about our research is that we use all modern technology of molecular, genetic, microscopy, but if I need to define a feature that is really kind of unified all of research is that we do look on comparative biology. We look on many organisms, and also in my presentation later you're going to see that we are shifting from many animals, and we try to compare between them, and we try to understand why changes happen along evolution, and then provide a molecular explanation for that. So that's kind of our approach. We would like to go look at many organisms. We like to do the very basic one. We also like to do and take it to the clinical and the human and the medical. So we are really kind of we try to get both benefits, the basic research, the interesting biology, but also the human connection.
Yeah, we saw your recent paper, which is pretty interesting. So can you please talk about it through your presentation? Thank you.
So, okay, so I will shift to my presentation then.
Yeah.
And you tell me if you see it. Do you see it now?
Yeah, go ahead.
Okay, very good. So this is really just kind of emphasizing again our approach from basic research to translational through a comparative approach. I will not repeat that. The kind of the basic question that we are trying to address right now in our research, because our research do move and change quite a lot through the last 10 years. But the current major question is, when we start our life, how does it the beginning happen? And we all know it starts with a sperm and an egg. So what is the relative contribution of those two structures, to those cells, to the first cells that is the beginning of our life, the zygote? And we all know that the DNA comes from both the mother and the father. But actually, there are many, many structures, and I think the most famous one is the mitochondria, that coming from the mother only. Most of what we have in the beginning to support life come from the mother. But there is one structure that is coming exclusively from the father, and this is this structure called the centriole. And let me just explain shortly what is a centriole. So here you see a picture with many organelles that a eukaryotic cell have, and we are going to focus on these two structures that are usually found at the center of the cell, at least in cells that during a certain stage. And these two structures have a very unique organization. They are made of 9 triplets of microtubules, and those microtubules are really the signature of this structure, at least from how people define them. And here you could see on the top a cross-section, what usually cell biology will use to ask the question, do I see a centriole? He will see these 9 triplet microtubules, and that will be the definitive element. Okay, I'm looking on a centriole. Centriole are quite big structure, like the size of 200 nanometer by half a micrometer. They're almost the size of a small bacteria. They are surrounded by this material here in dark, the PCM, which then act as a mechanism to nucleate astral microtubule, basically creating what we call a centrosome. So the cell usually have a centrosome, which acting as a microtubule organization center. The centriole does have a unique double life. They either form a centrosome like we saw here, but usually when a cell enters G1 in the cell cycle, the centriole will move to the surface of the cell, and the order of them will become then named basal body, will form a flagella or a cilia, which is very important for cell movement, but also for sensory function. Usually as the cell move on, the cilia will resorb and will regain the centrosome that usually function during cell division. The key thing that from this presentation, from these slides, to understand about what I'm going to say today, is that the cell dividing cell have two centrioles, and when I say two, I mean precisely two, not one and not three. And those centrioles are structurally defined based on the presence of microtubule, and they have this unique shape of a barrel shape. This is a typical centriole. What I'm going to show you soon is that there are actually exceptions to that. Because centriole number is so important, cell must have two centrioles, and the two centrioles are actually a little bit different from each other. We call them a mother and a daughter. These two centrioles need to duplicate, and then after the cell duplicate, then they are precisely segregated such each daughter cell have two. And this is the basic mechanism how our cells make sure that every cell have precisely two. So kind of the key question that we are asking is, okay, through our development, if you have a cell that have two, and then we develop it, the cell divide, become 4, they divide to 2, and this is happening again and again, that's all great. But how the beginning, how the zygote, how the first cell obtain its two centrioles? And this is not trivial. It's not trivial because one of the interesting phenomena is that, for example, the mature egg have no centrioles. The egg provides 0 centrioles. So how do we inherit the two centriole if the egg does not have it? The answer is really sperm. And here you could see a kind of the skeleton of an evolutionary tree, and you could see that many animals, many basal animals, have in their sperm two centrioles, which indicated here. And if you have two centriole in the sperm and a 0 centriole in the egg, you have two centriole and everything is okay. The zygote can start its life and everything moving ahead. But here's the problem. If you look on mammals, like humans, for example, we have only one recognizable centriole in the sperm. The same thing actually happens in insects. They have only one recognizable centriole based on structure. And there is actually something much more weird in mice, where you could see we cannot recognize any centriole. So the dogma is that there is no centrioles in the mice sperm. So how do we go from 1 plus 0 to a 2? That's basically the kind of a major question that we are asking those days. And just to reiterate the question, I want to mention a phenomena called centrosome reduction. And this phenomena is important because this is a phenomena that will take an egg precursor cell or germ cells, which have two centriole, and eliminate them. So the egg cells at the end of the process of maturation will have 0 centrioles. In the sperm, the common concept or dogma in the field is that you start with 2, like every other cell, then through the process of centrosome reduction, you lose one of the centrioles, and you're left with 1 centriole. And then somehow, in a mechanism that people propose, that maybe if we have 1 centriole know how to duplicate, then you will get 2 centrioles, and that solves the problem. But this idea actually was never tested. It was an idea that was accepted because it makes a lot of sense, but no direct evidence to that exists. And I'm going to basically challenge that idea. I'm going to propose an alternative mechanism. This all started actually about 9 years ago, when we have studied centrioles in fly. So that's so here you could see how really getting to the question from the basic mechanism. At that time, we were asking basic questions about how centriole are formed. We have identified then a protein called ANA1 GFP that was later named ANA1 by other one. It's not important. It's a centriole protein that labeled all centriole that was discovered just around that time. And this protein was fused to a green fluorescent protein, and you could see here the fly testis really labeling all the centriole. If we magnify and we look here on the area of the stem cell where the number 1 exists, you could see that there are 2 centriole in the cell. And one of the interesting things about the fly is that these centriole actually become very long, so we can actually see them very clearly. If we look on a sperm cells, for example, here you can see the nucleus in blue, the centriole in green, and the sperm tail in red. You could see that indeed there is 1 single centriole. This is post-meiosis. After meiosis, the early spermatid, the cell that will become the spermatozoa after differentiation, have 1 centriole. And this is what was known for decades by the work of many, many scientists. But one thing that we noticed is that, just a second, one thing that we noticed is that there is as we go during the development, we see in addition to this line a small bump. And then if we follow this bump a little bit later, it actually move away, and it is an own entity. And actually here you could see on the top, on the right corner with number 7, that every long centriole have this tiny thing. So what is this tiny thing? Just to kind of make the story short, this tiny thing is what we call an atypical centriole, which I'm going to explain a little bit. What do I mean by that? So we call it a centriole because if you look on a variety of centriolar protein, they are all labeling this entity. So this is a structure that, even though was not recognized before, it actually contains many of the centriole-specific proteins. When we do electron microscopy and on this centriole, we could see here, this is the regular centriole that have this 9 microtubules, but then you could see that there is this pericentriolar material, which is very dark in the insects. And then you could see this entity that does not have microtubules. And if you do a cross-section of that, you could see that it's actually made of an electron-dense wall with a central structure inside the central tube. And here on the side, you could see a microtubule, by the way, and you could see, compare the size. This is really a tiny structure. And here is a model of that centriole based on our structural studies. And just for comparison, here is a normal centriole. So a normal centriole is 200 nanometer by 500 nanometer. This structure is only 100 nanometer by 100 nanometer. It does not have microtubules. So this structure was really not observed in the past by people because it looks so differently. Also, beyond that, we needed to use really a special type of electron microscopy, high pressure freezing, free substitution, to be able to see it. Because when you do regular chemical fixation, it's very hard to see it. And I think that's the reason it was missed in the past. So, okay, so there is this entity. What is it doing? And we have followed it post cell division. So we fertilize sperm with an oocyte with a sperm, and then we looked what happened during meiosis. So this is this is the first cell, this is the zygote. You see here the DNA in the middle, you see the spinal microtubule, and you see a beautiful centrosome in both sides. If we focus on the center of one of them, you could see here the long centriole, and you could see actually another dotted centriole nearby. We know this is a dotted centriole that was formed in the egg because the way the experiment is constructed is that we are using an antibody that is labeling a protein expressed from the mother side. And we can see on the other side that there is 2 small centrioles. One of them is labeled by green. And this is again because the way we constructed it is that we are labeling a protein, a green fluorescent protein that is coming from the mother side, and we can follow it. So these 2 purple structures coming from the father, one is a very long one, look at centriole, and we presumably presume that the other one is the atypical centriole, and these 2 new entities are really a dotted centriole formed in the zygote. So that support the idea that the sperm provide 2 structures, a centriole and then this atypical centriole. Next, we wanted to ask the question, what is the function of this atypical centriole? And what we have done a genetic screen to identify mutants that have defect in this atypical centriole. So here you could see a regular centriole when we label it by again with this protein ANA1, we have the regular centriole and the atypical centriole. And then we have identified a mutant that have only the regular centriole, but the atypical centriole is largely affected and it's very hard to see it. So this was a defective atypical centriole. What we can do now, we can take these 2 flies, each in a time, and fertilize a normal egg and ask what is happening. So when you use a normal male, as I showed you before, you're going to get in the zygote 2 centrioles on each side and DNA in the middle. And however, if you fertilize with this fly that have no atypical centriole, we get many times monopolar spindles. And the other pole is missing. This suggests that the atypical centriole is actually essential and function during the first zygotic division. After doing those studies that basically demonstrated that in the fly there is this atypical centriole structure, and this atypical centriole structure is inherited to the zygote and is essential in the zygote, we want to ask the question: is this a general thing, or is this just a fly thing? And Here you could see a small study that we have done in beetles. And when we have, beetles was thought for a long time to have only one centriole. But when we stain again with this antibody against this ANA1 protein, we suddenly see a second centriolar structure, atypical structure. And we actually can do EM and show it have a very similar dimension as the atypical centriole of the fly. It's 100 nanometer by 100 nanometer by electron microscopy. So this led us to the hypothesis that in addition to the typical centriole, insects in general provide also a second atypical centriole that look very different, but it can do what the centriole do and substitute for it. So the next question, after we have done this basic research in the fly, and we are continuing asking many mechanistic questions, how the atypical centriole is formed, and how does it function if it's so different. One thing that we wanted to ask is, okay, we've just shown here on the left side that the flies and the beetle have actually two centrioles, very much like the main core of the evolutionary tree. Well, then one would hypothesize that maybe there is also atypical centriole in each one of us. Maybe our sperm, the human sperm, the mammal sperm, have one centriole we recognize and another one that is waiting to be identified. So we have went forward to ask this question. But first I want to introduce you what was the dogma in the field. What people believe before that. So as I told you, every cell starts its life, or almost every cell starts with two centrioles. In the mammalian system, we call one centriole that near the nucleus proximal centriole, PC, and the one that is further away, we call distal centriole. And the distal centriole is the one that will form the flagella. And it is attached to it. It's physically attached, templating the formation of the flagella. If you look on a mature sperm, not an early spermatid, but a mature spermatozoon, you'll see only the proximal centriole. And this absence of this other ability to see the other side led to the conclusion that there's a centrosome reduction and we have only one centriole. Here you could see, for example, a published work that depict this idea. Here's the nucleus on the top with the proximal centriole here, and you could see that here there is this white area with no centriole. So that's what created the idea that there is only one centriole, and this distal centriole is reduced to a remnant or to a nothing and does not function post-fertilization. So the first thing that we have tried to do was, the first thing we have done is basically contact many of our colleagues, ask them, give us tools to look on centrioles. Any protein that was available to any of our colleagues, we reached out, and everybody was very nice and gave us those tools. And I'm going to show you just two categories of our results. Some categories, like in this case, you could see the nucleus is an ejaculated human spermatozoa. You could see that there is one centriole. And this is exactly what the dogma said. The dogma said there is only one centriole. And that's what people have seen before, and that's why they believe there is only one centriole. But if you look on other proteins, like, for example, centrin, you see clearly two dots. One colocalized with the proximal centriole, and one more further away, which will fit the category of a distal centriole. Actually, we find some proteins that are enriched in this distal centriole. And we actually, for POC1B, we checked 3 independent antibodies, come from 3 different sources. 2 of them we generated ourselves. And we always see more POC1B in this other centriole. And just for a shortcut, we have done it with many other proteins. We have about 9 proteins now that demonstrate that there is these second entities here. The next question was to ask, where is this centriole? Where is this other thing? Because here you could see an EM again showing the proximal centriole and this empty space we call vault. And we have collaborated with Jandranka from NIH, and she do a beautiful correlative light electron microscopy. And when she did that, and she placed the POC1B GFP signal, you could see that it labeling one of them, what you expect, the proximal centriole, but the other labeling actually labeled below this empty space. It labeling the tip of the axoneme. And you could actually see that the microtubules here are actually changing their morphology. So what we conclude from that is that this atypical or distal centriole is found connected to the base of the axoneme and not where people expected it to see it in this empty area. So we again went to do this high pressure free substitution electron microscopy because we know it gives us a much better picture. And when we do that, you could see here the microtubule. This is a longitudinal section. And you could see here the microtubule. This is the axoneme. But then you could see how they splay up. And the white arrow here show you how it's open and become much more wide. And if we do a cross section at that level here, you could see that there are microtubules, but they are not organized in a 9-fold microtubule anymore. So what we have here is, again, is a atypical organization of microtubules. They are completely distorted from normal organization, but they are there. And actually, we can see them with any antibody that we have used. We use about 4 different antibodies against microtubules, and we always see them here. This can be seen much more clearly maybe in this picture, which is a super resolution microscopy of this structure. Again, it's ejaculated human spermatozoa done in collaboration with Vito Menila in the University of Toronto. And you could see here again the proximal centriole, that area. You could see the axoneme, and then you could see those splaying microtubules. The interesting thing is that when you look on this protein, in this case, I think it's POC1B, but we have seen it with many other proteins. We see these 2 bars that are flanking. You see the microtubules, and they are flanking them from the inside. I'm not going to show you, but we have done also storm super resolution microscopy, and we see a similar results. We have done it also in the bovine in this case. So this atypical centriole have a very different organization. And here is a model of this atypical centriole. So here just the proximal centriole that maintain the same organization. I just want to highlight that even though it have the same organization, it is atypical in its composition. So it's not a normal centriole in that aspect, but structurally it's look very much like what we expect, and people can see it. But the reason the atypical centriole, the distal centriole, was not observed is because you have this unique organization where the microtubules are disorganized, actually very hard to see them, and they are surrounding this vault area, and there are these 2 rods made of POC1B, 17, POC5, and we have another protein now that we identify that label this structure, and there is this other protein that label kind of the top. So here we have another atypical centriole that exists in the human sperm and also in other mammals like bovine and rabbits. The next question was, what is the function of this guy? Is it really working? And here we have done a collaboration with Ann Miller, that is an expert with frog extract. You know, the difficulty that we have is that there are some legal and ethical consideration when you do experiment with human. So we were very careful to follow all those things. And what we have decided to do is to take a egg extract from frogs and make a big, like a shake out of it. So we have basically just protein. We separate the DNA from the proteins, and we have only the proteins here. And then we take a human sperm, and we use detergent to strip the membrane out. So it's not really alive anymore. But we don't kill the protein inside, and then we can expose it and ask what is happening. And here you could see this sperm just without, if we do not expose it to extract, you could see the nucleus here, you could see the proximal centriole, that this is found just at the. If we now expose it to the extract, we see that the tip of the axoneme recruit gamma tubulin. And gamma tubulin is a marker for the pericentriolar material. So this is basically mimicking, it's an in vivo assay, a functional assay to ask, can this distal atypical centriole do what a normal centriole do in an egg? Can it recruit pericentriolar protein, PCM protein like gamma tubulin, which then form a microtubule ester? And as you could see, actually we see much more recruited to the distal centriole than to the proximal centriole. So that was our first hint that this atypical centriole is actually functional. The next thing we wanted to do is something in vivo. And to do that, we have collaborated with 2 great scientists, that really one of the leaders in the field that came up the idea of centrosome reduction, Joey Sherton and Kelvin Simmerly from the University of Pennsylvania. And what we have decided to do is to study it in the bovine, because bovine, very much like human, is thought to have the same centriole inheritance. And you could see here that when we fertilize an egg with a bovine sperm, we can now stain it. And you could see here the 2 nuclei, and you could see here the axoneme that is labeling the base of attached somehow to the nucleus. And let's zoom on it. And when we zoom on this area, we could see that the axoneme, which is the flagella of the sperm, at the base actually recruit CEP152. CEP152 is one of the proteins that get eliminated during the centrosome reduction, the remodeling process. And now it is being recruited from the egg to label it. And you could see it here also. Not only that this we see it during interphase, we see the same thing during mitosis. So here you could see a beautiful picture that of a zygote in mitosis. You have a spindle, microtubule spindle, DNA in the middle, and now we can zoom on, we can look on the centrosome on one side, the centrosome on the other side. First thing you could see that the centrosome on this side actually is connected to the axoneme of the sperm tail. And that's a sign that we actually have here the atypical centriole functioning. And here you could see a magnification of that, the axoneme labeling and the base of the centriole. So the base of the axoneme where the atypical centriole is present is actually functioning by recruiting PCM and localizing and forming. So basically, just kind of summarize our finding, this atypical centriole can recruit PCM like CEP152. It can form an ester like you could see here around. I did not show you, but and he can reside to the pole. And actually, I did not speak about it. It can also direct the formation of a new centriole. So this atypical centriole behave pretty much like a regular centriole. It's just very different. So to conclude our studies up to now, we think that across evolution, with the exception of mice that we have not yet looked on in detail, there are two centrioles, either two normal-looking centrioles, like in the most basal animals, but in insect there are two, because there is one typical and one atypical, and also in non-rodents mammals have one typical and one atypical. The interesting thing is that the structure is very different, and of course we are very interested in the future to understand why this centriole become atypical, and how they become atypical. Because, for example, one can think that if there is a defect in the mechanism that makes the centriole atypical, this may lead to infertility. And we think actually that this will lead to unexplained infertility, because our prediction is that the sperm will look pretty much normal. And if the sperm is normal and you go to the physician, well, nothing is wrong with you, but maybe there is something wrong with you. And that can be an unexplained infertility. So we have several ideas that we are trying to explore with this new structure. So just to summarize the new model that we have for centriole inheritance. So centrosome reduction happened in the germline, but instead of reduction in the male, we have remodeling. One of the centriole become atypical centriole. In the egg, yes, as much as we understand, the two typical centriole disappear. These two typical centriole from the male are inherited to the zygote, and they form, each of them form an ester, and then they help in the migration and localization of the two pole nuclei to form a mitotic spindle. Each one of them go to a different pole. One pole will have the regular centriole. By then he will duplicate and will form a normal centriole, which we call it zygotic dotted centriole, but it will be a normal typical centriole. And on the other side, the atypical centriole will do the same thing and will have this zygotic dotted centriole. And that's, by this way, we restore the normal complement of centriole we expect in a cell. This was funded by different organizations, and throughout the lecture, hopefully you saw the people that contributed to the different aspects. This really was a work done by PhD students and postdocs in the lab, and also by many undergrad. Here you could see Rachel Roifman, for example, an example of an undergrad that was in the lab, and many other undergrad. And we really enjoy the enormous interaction with many colleagues in our field. This is really very fascinating field, reproductive biology. People are very collaborative, and they help us, and we are continuing this kind of a direction. Thank you.
Thank you so much. So can you please? Okay.
Yes.
Okay, here you go. So it's a wonderful talk. Thank you very much. I really appreciate that. I don't know if any viewers have any comments, questions, or anything. They can type it up, basically. I'm just trying to see if, yeah, there isn't anything. So, I mean, like you have explained it in pretty nicely, the way it happens and all those things. So this may be in the future. I have one question. Like, once you have these centrioles, right, like after meiosis and all, they move to the other side. So you showed in the picture that the atypical one moves. Is it sure that atypical one only moves to the other side, or like is there a specific way of they are moving?
So you're speaking about the fly situation. In
Yeah.
the fly, there is a reorganization of the area, yeah, I think this is, I don't know if you noticed that this is the location of the centriole is species specific. So in the fly, they're organized the way like that, but in the beetle, they're actually one on top of each other. And if
Okay.
you go through different on the phylogenetic tree of animals, really the position of the centriole is highly variable. What is not variable is that there is always two. It's also highly variable how they look. So there is a conservation of number, but not structure and not position. And that's really fit very well with the knowledge about what is the role of centrioles. Centrioles really need to maintain number. That's a critical thing. And as we get more knowledge about what is the structure of the centriole do, then we understand, well, maybe it's not critical at a certain stage of the cell life to have a typical centriole. And I think this is really one of the impact of this work is that I think people should consider that in certain cell type, especially differentiated cell type, in a certain cellular stages, maybe the centriole look a little bit different from what we expect. And it's for a purpose, for some biology that we need to discover.
And in the atypical centriole, there is those microtubules, right? So they look different, disorganized. So do you know why and when it becomes again reorganized and all?
So that's exactly what we are doing right now. We have studies in the fly. We are trying to understand the mechanism. And we have also studying in the mammalian system. And we have just discovered some key player, we think, that actually stabilize homeogenesis. It's important to understand that when the centriole, the cell, the spermatid cell is round, it transitions into this beautiful, like moving sperm. During that time, there are many, many days of transformation. And during that time, there is a major transformation in the centrioles. It's a multi-step process that we can actually image it, we can see it. We see enrichment of protein, then they are redistributed. So this is exactly what we are studying right now. We think this is very important. We think this can explain, I said already spoke about infertility, but I can tell you that there are other issues like spontaneous abortions that happened that may be explained by that, because everybody thinks the embryo is okay, but in the absence of atypical centriole, the embryo is really not okay, and things start to go wrong, and then the embryo will not develop normally. And so we are trying to understand the mechanism to find the role. We think that part of it is really key protein that are being changed, and there is a new set of protein in the sperm. In the fly, for example, I can tell you, we have shown that there is a protein that have a different two protein isoform. One isoform go to the atypical centriole, and another splice isoform go to the typical centriole. So it's the same gene, but they have two products. One go to the atypical centriole, one go to the typical. So we think this concept is maybe involved in the generation of this diversity of centriolar structure.
Thank you so much. I really appreciate it. It is wonderful, fascinating work, and I look forward to more of your papers in the near future.
Thank you very much for this opportunity.
So you can share the link with anybody you want to. It will be on our website. So good luck again. Thank you.
Thank you.